The most remarkable property of living cells is their ability to produce exact replicas of themselves. For today's discussion, we will be discussing all about nucleic acids. Two types of nucleic acids are found within cells of higher organisms. We have deoxyribonucleic acid, or also known as DNA, and ribonucleic acid, or also known as RNA. Nearly all the DNA is found within the cell nucleus. Its primary function is the storage and transfer of genetic information. This information is used to control many functions of a living cell. In addition, DNA is passed from existing cells to new cells during the cell division, while RNA occurs in all parts of the cell. It functions primarily in synthesis of proteins, the molecules that carry out essential cellular functions. DNA controls all living processes, including cell division. It also carries the genetic code. DNA is the molecule that makes up chromosomes. The model of the DNA was developed by Watson and Crick in the year 1953. They received a Nobel Prize in 1962 for their work. The model looks like a twisted ladder, or what we call the double helix. As mentioned earlier, DNA is present in all cells and virtually restricted to the nucleus. The amount of DNA in somatic cells of any given species is constant, while the DNA content of gametes is half of the somatic cells. In cases of polyploidy, the DNA content increases by a proportional factor. RNA is a messenger that allows the instruction of the DNA to be delivered to the rest of the cell. Let's have a recap of the distribution of the nucleic acid inside a cell. Take note that DNA is found in the nucleus, with small amounts in the mitochondria and chloroplast, while RNA is found all throughout the cell. All nucleic acid molecules are polymers. A nucleic acid is a polymer in which the monomer units are nucleotides. A nucleotide is a 3-subunit molecule in which a pentose sugar is bonded to both a phosphate group and a nitrogen-containing heterocyclic base. With a 3-subunit structure, nucleotides are more complex monomers than the monosaccharide of polysaccharides and the amino acid of proteins. Nucleotide serves as the building block for DNA and RNA. It also functions as the intracellular source of energy, or the ATP. They are also involved in intracellular signaling and intracellular signaling switches. This is the block structural diagram for a nucleotide. All nucleotides are two-ring structures composed of 5-carbon sugar, may it either be a beta-D ribose for RNA and a beta-D2 deoxyribose for DNA. It also has nitrogen-containing heterocyclic bases. We have the purine and pyrimidine. And the third subunit is the phosphate group. The sugar unit of a nucleotide is either the pentose ribose or the pentose 2-deoxyribose. Structurally, the only difference between these two sugars occurs at the 2'. The hydroxyl group present on this carbon in ribose becomes a hydrogen atom in 2-deoxyribose. Take note that the prefix deoxy means without oxygen. Take note of the numbering system for this pentose sugar. Pentose ring atoms are designated with primed numbers. RNA and DNA differ in the identity of the sugar unit in their nucleotides. In RNA, the sugar unit is ribose, hence the R in RNA, and in DNA, the sugar unit is 2'-deoxyribose, hence the D in DNA. The next subunit is the nitrogen-containing heterocyclic bases. 5 nitrogen-containing heterocyclic bases are nucleotide components. 2 are derivatives of purine, a bicyclic base with fused 5 and 6 members rings. Now, for the numbering system of atoms of nitrogen-containing base ring atoms, they are designated with unprimed numbers. Adenine is the 6 amino derivative of purine, and guanine is the 2 amino 6-oxopurine derivative. The next three bases are derivatives of pyrimidine, a monocyclic base with a 6-membered ring. Thymine is a 5-methyl 2,4-dioxo derivative. Cytosine is a 4-amino 2-oxo derivative. Take note that thymine is only found in DNA. In RNA, thymine is replaced by uracil. Uracil and thymine are structurally similar. This is the summary of the occurrences of nitrogen-containing heterocyclic bases in nucleic acids. Both purine bases, adenine and guanine, are found in both DNA and RNA. For the pyrimidine derivative bases, cytosine is the only base found in both DNA and RNA, while thymine is only found in the DNA and uracil is only found in RNA. Phosphate, the third component of nucleotide, is derived from phosphoric acid. Phosphate groups are what makes a nucleoside a nucleotide. Phosphate groups are essential for nucleotide polymerization. Illustrated below is the basic structure for the phosphate group. The number of phosphate groups determines nomenclature. For instance, a monophosphate, for example, adenosine monophosphate, its free form is an inorganic phosphate. Diphosphate, for example, we have adenosine diphosphate. We have here two phosphate group whose free form is pyrophosphate. And the unit that we use for energy, adenosine triphosphate, is an example of a triphosphate, wherein we have three phosphate group. It does not have a free form.